Hello, everybody. Thank you for joining our webinar today on infection control and specifically decontamination of surface and air is what we're going to talk about. First of all, I am disclosing that I am an employee of Health First, and they're the sponsors of this course. My name is Dwight Shreve. I am out of the Houston, Texas area, and we also have Dr. Don Cohen with us. He is our Chief Clinical Officer. He's also on the board of directors of OSAP, and he's also on our medical advisory board. So he'll be joining me today, and we'll be discussing a lot of things when it comes to air decontamination and surface disinfection. So a couple of housekeeping things I just want to kind of start out with. Just want to let everyone know that you're going to receive an email by the end of the week with a link for you to be able to go out there and get your CE. Everyone will receive a copy of the slides that we're going to go over today. We're going to, there's a lot of slides, there's a lot of information. We want to make this more informational. I'd rather you guys kind of listen and engage with us and not really so much worry about the slides and concentrate on them right now. We will address them, but like I said, you guys will get a copy of this. So sit back, relax, and we can have a good time here. If anyone has any questions during the presentation, feel free to throw them in the Q&A, and we'll try to address them throughout the presentation if we can. If for some reason we can't get to it, we'll definitely follow up with you, because we do want to make sure that all your questions get answered. And then, again, we want to make this kind of engaging and interactive. I know it's a little bit hard to do in the virtual world, but we'll be asking you guys some questions, and you can throw those in the chat, but we'll get to those throughout the presentation. So let's go ahead and get started. This is kind of what we're going to try to learn today, the objectives that you can see there on your screen. Again, we're really focusing on contamination of surfaces. We're going to talk about CDC recommendations, hopefully give you guys some tips and guidance, maybe some refreshers on those protocols. We're going to talk about some aerosols and methods recommended to reduce the risk of aerosols and some adjunctive technologies. So first off, let's start about, kind of get down to ground level. We talk about the chain of infection. One of the things that we try to do is break that chain. When you look at the chain of infection, you think about infection and transmission of these microorganisms. I mean, first of all, there needs to be a pathogen. There has to be a sufficient level of it to cause a disease. First off, there's got to be a source or a reservoir. There's got to be a mode of transmission or an entry route, also known as a portal, and a susceptible host.
Hey, so Dwight, here's the key. We are the susceptible hosts. So anything and everything we can do to decrease that susceptibility, and that's when we're talking about things like the CDC and layering our PPE. Why do we wear it? What levels are we wearing? Really, really, really important. It's through that we can truly break the chain. Thanks, Dwight.
Yeah, absolutely. No, feel free to jump in, Dr. Don, whenever you want. We have, in order to break that chain, we at least have to break one of those links, right? Somewhere in that chain, we've got to break something down, so that way we can protect ourselves, protect our patients, and reduce the risk of infection. So kind of going to jump into the contamination of surfaces here as this slide. We kind of break it down into two, or CDC breaks it down into two categories: housekeeping surfaces and then the clinical contact surfaces. Dr. Don, what are some housekeeping surfaces that we could talk about?
Well, when we talk about housekeeping, we could be talking obviously about the walls, the floor. We could be talking about almost anything. And we normally think about the surfaces like our tray tables or the counters in the operatory, but be reminded that, oh, well, the floors are really not the thing, or the walls. And there it is with Mrs. Jones in the chair, and she has her grandson crawling around on the floor. So nothing is eliminated from any of these pictures. Dwight?
Yes. Thanks, Dr. Don. Yeah, I was thinking about that, because when we talk about the things that you mentioned, floors and walls and blinds or such, we don't really think about cross contamination coming from the floor unless we're doing handstands, right? But to your point, I didn't even think about that. If you got babies crawling around in your offices, yeah, they're picking that stuff up, and you know what they're doing, what kids and babies love to do, right? They love to touch a lot of different things all over the place. The coffee tables or the armrests in a chair. So no, I appreciate you bringing that up. I didn't even cross my mind with that. So we talk about how long can it live on a surface under ideal conditions, right? There's been more focus on the length of time a microorganism may survive on a surface, especially since the pandemic. And I think it's very important to note that, not all microorganisms are built the same, right? So some are more resistant to others. Some can survive longer than others. What was astonishing to me, you look at MRSA, for example, 7 days to 7 months that can stay alive. Or Mycobacterium tuberculosis, like TB, 6 to 8 months. That stuff can be hanging around in certain areas if they're not properly cleaned and disinfected, right? So, it's just kind of striking to see that, HIV, remember when HIV was huge, still is, but, when it first came out, we focused on that quite a bit. Do you remember those days, Dr. Don?
No, that was before I was born, actually. I'd like to say that. The reality for all of us on looking at this is, don't only think about your operatory and don't only think about your offices. Think about anywhere else. Why should we constantly be washing our hands? Why is it important? Because any place you touch could very well be a contaminated surface. HIV was the easiest of all because a dilute 10% bleach solution would do the job. Not for the rest of these, and especially when we're talking about things like TB. We've been lucky so far. MRSA has not become an issue in dental offices, but same thing for C. difficile, not so much yet, but nothing is safe. And as Dwight pointed out, when we think of COVID, look at that 2 to 3 days. Don't stop washing your hands.
Yeah, and some of the variables, right, when we talk about the pathogenic or these potentially pathogenic microorganisms, we look at their modes of transmission, what it is or what they are, their survivability on the surfaces, the resistance to kill by chemicals or even heat sterilization, the dose needed to transmit disease, and how quickly and how severely ill a microorganism may cause an individual to be. So let's look at some of the, resistance of microorganisms here. What's kind of one thing that kind of shouts out at you on this slide, Dr. Don?
Well, the reality is we need to think of which organisms are the easiest to kill and which aren't. And as we think about enveloped lipid viruses, and you can see those are guys that we know: HIV, HBV, HCV, and coronaviruses. And they're relatively easy to kill, relative, but that's a relative term as well. We need to do the right things. If we do the right things, and if we're using the right chemistry and using the chemistry properly and cleaning first, we're in pretty good shape. And most of us have been trained fairly well in our offices on how to do that.
Yeah, what stuck out to me was bacterial spores. That's the most resistant, and look at the level of resistance and what it takes to actually kill those. I mean, that's your sterilization, right? You got your different levels of disinfection from low, intermediate to high, and then you go into sterilization, and that's what we're doing to kill those spores. And then you look at TB, which is going to require like an intermediate level of disinfection. And so, that's what I love about the slide. It kind of breaks that down so that we can kind of get an idea of what it's going to take. You know, one thing it talks about in this slide here a little bit is what we call enveloped or lipid viruses. Those are going to include the HIV, your Hep B, Hep C, that's your coronaviruses, right? The least resistant or are the enveloped or the lipid ones. Those are medium-sized viruses. And look at the next slide here. So we'll talk about some clinical contact surfaces and some CDC guidelines. You know, it's very important that we use hospital-grade disinfectants that are the EPA, they're EPA registered on the list N. If you don't know if your products are on that list N, I would highly recommend you contact your dealer or distributor, or probably would be better is actually go on the EPA website, Google that EPA list N, and find out if what you're using is on that. You know, running to Walmart and grabbing some Lysol that would use in your home, it's not hospital grade. You know, that's not a good idea, not what we want to do.
And you know what, Dwight, I'm going to jump in because this is something you pointed out earlier. And there are two words here that keep coming up: clean and disinfect. And I'm going to steal some of Dwight's thunder, and that is, we all know we're supposed to clean. We all know we're supposed to disinfect. So the chat question I would ask everybody is, can one wipe take care of all of that? And I'd ask that you respond on chat. And the reasoning we do that, I'm going to leave more to Dwight, but that's a key element of everything we're doing, is this issue of clean and disinfect. Dwight?
Yeah, that's a good question. You know, when we talk about even the CDC here, it talks about on the slide is cleaning and disinfecting, right? How often should we be doing it? When should it be done? Where should it be done? I mean, those are kind of some of the things we're going to kind of dive in today, because I think we'll probably bring to light a lot of areas of opportunity that we didn't know about. But let's see here, some people. A lot of people, yeah. Thank you guys for jumping in. So, you know, so far the answer has been no. There's not one that does it all. So, you know, I know you weren't born then, Dr. Don, but I'm kind of jump back a little bit and into the 80s. You know, I did probably anywhere from 80 to 100 audits last year in dental practices, and you know, at their request, just helping them look at their protocols and observing and whatnot, and then doing some teaching. And one of the things I would always talk about is, as you mentioned, it's a cleaning and disinfection. It's a two-step process, right? We're going to do one to clean, so that's your wax on, and one to disinfect, that's your wax off, right? So it is a two-step process, and I will tell you that in every one of those offices, I didn't observe that. And it may be because of what the wipe they were using, but as people were pointing out here in the chat, there's not really a wipe that's going to do both, right? But what I want to talk about is the importance of that. So when we look at surfaces, when we look at these clinical contact surfaces, I mean, if there's visible soil that's on these surfaces, obviously, you know, we can visually see that. We need to clean it. We need to wipe all that off. But then there's a lot of times where we can't see what's on there, right? Just because it's not visible doesn't mean it's not there. So making sure that we clean that surface first, and then applying the disinfectant on top of that, because you cannot disinfect what's dirty, right? And you brought up earlier, we were talking this morning, instrument reprocessing. Remember that story you were telling me?
Yeah. So that? here's the one. This has killed me throughout my practice. I would be either applying some composite or maybe some cement or something, and there's somebody handing me a plastic instrument, and there's dried up cement on it. Now, they just took it out of a pouch that came out of the autoclave. So I know it's sterile, but it's dirty. And to Dwight's point, that's what this is about. Everything has to be cleaned of soil. It's bad enough that we can't really detect with our eyes if there is any form of bacterial contamination, but certainly soil Yes, and therefore it's imperative that we use that two-step process.
Yeah, because if that soil is covering it, then that disinfectant can't work through it, right? It can't get to what's underneath there and kill anything that's going on. So, just to kind of to the slide here, again, it's just kind of reiterating what we talked about: hospital level disinfectant, EPA list in, two-step process. And we talked about the intermediate level disinfectants and whatnot.
As we go to the next slide, the key here, as it says, key steps for success. I think one of the most important things that I could ever think of is if you don't follow, and Dwight, you brought this up this morning, if you don't follow the IFUs on your products that you're using, and that would be the instruments, could even be the machines that you're using, IFU, instructions for use, you will mess it up. And the reality there is, for everybody on this call, not a poll question as much as, do you really know how many minutes it takes for you to truly decontaminate a surface? In other words, you cleaned it and then you're going to wipe it down. How many minutes does it take? Because if you can't just spout that answer in a heartbeat, you best believe that the rest of your team members or staff aren't going to be doing that either. And that's scary.
Yeah, I tell you, that is one of the things when I do walk into offices, and I am doing an inspection or an audit or whatnot, that's one of the first questions I ask is, hey, what wipes are you using? And a lot of times the answer is either, Sandy cloth or cavi wipes. And then my next question is, what the kill time is on it? And a lot of times I get the deer light, the deer in the headlights there. Like, well, what do you mean? So, instructions for use are so important for several reasons. And I just want to touch on that real quick. Everything in your office is designed to work and do a certain thing or a certain task, right? And in order for it to do what you want it to do, the manufacturers put together these instructions. So we need to read these instructions so that we know that it's going to do what we want it to do. First of all, then what's the point, right? Secondly, we look at a liability issue. So when you have a product or whatever it may be, and you have a failure, if you followed those instructions for use as set out by the manufacturer, and there was some sort of failure that happened, well, then the manufacturer is on the hook for that. They're liable for that. But if we're not following those instructions for use and we have a failure, then we've shifted that liability off of the manufacturer now onto the practice. So that's why it's also, again, so important to follow those IFUs, because we want to make sure it's doing what it's supposed to do, but there's also that liability standpoint. We came up with an example this morning, Dr. Don. You remember what it was when we were talking about, where if that were to happen, one thing I know we didn't talk about this morning, but I think about dental unit water lines. I have a strong background in dental unit water lines. And more often than not, a lot of people weren't following the IFUs if they were using tablets to treat the dental unit water lines, i.e. they weren't shocking periodically and they weren't testing periodically. So let's say you have somebody who came in that had got infected, right? For whatever reason, they were infected through their dental unit water line because the CFUs were too high. If they were to be asked that question and asked for those results, right? How often are you guys shocking? Do you have that documented? How often are you guys testing? Can we see those test results? And you don't have that. Well, that's not on the manufacturer anymore. That's coming back on the practice, because they can't document and show that they were actually following those instructions for use, and that's why they had a failure to begin with. So, yeah, IFUs are definitely very important. And
while we're talking about that, if we remember on previous slide, although this is fine, recognize if the wipe you're using is not wet, it's not working. The kill times are all based on wet. So if you're using a disinfectant that is heavily alcohol based, either you better be working real fast, or you may be using multiple wipes. Likewise, if you're using a very small wipe, you may find that you're going to have to use a second wipe as well. But everything is based upon the surface being wet in order to have a true kill time.
And, thank you for bringing that up, Dr. Don, because it made me think, when you're looking at those IFUs, keep reading them. And I'll give you a prime example. Cavi wipes, right? Cavi wipes actually list 2 different kill times. I think let's throw this in the chat real quick. Which kill time do we use? What is it based off of? What do we base kill times off of?
And Dwight, that's a great question, because as we saw in the previous slide back when we were talking about resistance, there are certain organisms that are much easier to kill. HIV, we were just talking about. But there's a few others that are a heck of a lot harder to kill. They develop spores, and yet those are still in our dental practice. We need to be addressing those. So any of those results out yet, Dwight?
I don't see any, no. Come on, somebody knows the answer out there. What do we base the kill times on? That's the question.
What's the easiest? What's the hardest? So I'll sneak a thing out there. Obviously, things that produce spores, bacteria that produce spores are a whole heck of a lot harder to kill. And the one I always think of the most is TB.
TB, that's right. A lot of times we base kill times off of TB. So again, when you look at those instructions, you'll see 2 minutes for a lot of microorganisms. You got to keep reading to see TB is going to be 3 minutes. And different products have different kill times. I know Optum 33, I believe, has 5-minute kill time. I've seen 10-minute kill times on products. And what you were talking about earlier, I've seen 1-minute kill times on products. Those are the ones that are higher alcohol base. So the challenge there is keeping it wet the entire time. And so keep that in mind. That surface has to remain wet at room temperature for the entire time. And if you have to apply a little bit more, then so be it. But make sure that if it's a 3-minute kill time based on TB, that surface is wet at least 3 minutes. And then don't wipe it after the 3 minutes, just let it air dry.
So Dwight, I've got the question for you. So we did all of that. We used an end list. We used it for the 2 minutes or 3 minutes, whatever. How do we truly know? How do we assess that we actually have disinfected that surface?
That's a great question, Dr. Don. I mean, that is one of the challenges, right? How do we know if we're getting all of the surfaces? Did we miss anything? How do we know that what we're doing is actually working? Because, a lot of times we can't see it, right? Unless it's just visibly soil, we don't know, because a lot of these microorganisms are invisible to the naked eye. So what we talk about is some different methods that we can use to observe. Obviously, visual inspection is one of them. Again, we can look at a surface, and if it's visibly soil, we'll both probably agree that that's a dirty surface, right? But if it's not, and I look at it and I go, that looks clean to me, and then you come in behind me and goes, no, that looks dirty to me. The problem is that visual inspection is very subjective, right? You could have your opinion, I could have my opinion, we may not agree. So in order to kind of take that out, and let's have a more objective approach, is the use of ATP meters. ATP is, as you see in the slide here, it's produced by all living things. And a lot of times, we don't know what we don't know. And one of the things I learned early on, a phrase I learned early on in my career when I first got out of college, is what gets measured gets done, right? So we want to be able to take some measurements. We want some objective results. We can establish that by ATP meters. Now, it's not really used a whole lot in the dental settings, at least not yet. We do see these a lot in hospital settings. I've seen them used in restaurants. And what we do there, you have an ATP meter, and then you take a swab, and you're going to swab an area. Let's say 4 inches by 4 inches is usually a good area, and we're going to swab that area. All right, and then we're going to put that into the ATP meter, and the results are going to be in RLUs, which is what we call relative light units. So those microorganisms, they're bioluminescent, they're going to light up in that meter, and it's going to measure how many things light up. So if you kind of think, you think about these fireflies that are, lighting up, it's kind of the same concept there, right? You can usually get results in 15 seconds, so it's super fast, and it's measurable, and it's objective, right? It's there in front of you. So, we talked about that earlier, Dr. Don. We were talking about hospitals a little bit different, right? Because of HAIs. Can you talk a little bit more about that?
So,
understand.
So without a doubt, in dentistry, we don't have a specific number that we are forced to report back to any organizations. Hospitals, totally different. Hospitals actually have something called HAIs, hospital acquired infections. And hospitals are actually measured on those. And if they come above a certain median point, they actually get dinged. Their reimbursement levels can be dinged to a point of 1%, 2%, which when you're talking about billion-dollar organizations, that actually adds up to some money. And the way they test is using things like ATP meters. What that will actually do is wipe down 7 different surfaces in a location. And one of them may be the doorknob, another one, the counter, the screen, the bed itself. But the reality is they find things that not just this issue of ATP meters and swabs, but they'll go to a point. They did one, we were looking at a study where the ventilators, we know what a ventilator is because that's the ultimate problem of COVID when your lungs start to fail. Well, they found that the ventilators in a room had the highest level of RLUs, of relative light units. And you would look and say, but that's such an important device. The other thing that comes into this is called protocols. So that is a medical device. The cleaning team who comes in and does all of these surfaces knows to take care of everything in there. But on their list, they would never touch a medical device because in their mind, the nurses would be handling all of the medical devices. But the nurses on their side, oh, well, if it's cleaning, then it's obviously part of housekeeping. And it turned out that those ventilators had the very highest numbers by orders of magnitude. So when we get down to this, the interesting thing about using these meters or any other assessment device is it can actually help improve our protocols, because it is through the protocols, through our SOPs, that we truly will get the results that we're looking for. Dwight?
Yeah, absolutely. I appreciate that, Dr. John. I was thinking when you were talking about that story, I was thinking I was actually in a pedo practice I think it was last week or the week before, and we were doing a video shoot there. And anyway, one of the things I wanted to do is I wanted to take ATP meter readings, right? And aside from the operatory, because we know that gets cleaned quite regularly, but I wanted to look at the bathroom faucets. I wanted to look at the door handles. I know we did the door handle to the front door to get in, the door handle when they let you back into the operatories. But there was also a video game that was there. And I know since COVID, not so much, but every practice is going to be different and what protocols are following. But there was this video game, and I'm like, we have to do that video game. And I mean, without getting too much into the details right now in this presentation, but the RLU reading was about 850. 50 would be good, 30 would be better. So obviously the lower the better. But 850 is just kind of off the charts, right? There's a lot of cross contamination that's happening. And again, we got kids who are walking around and touching things, and I wanted to do the armchairs, but they didn't have any armrests on their chairs in the reception area, because I know that's also a high touch surface area. So you think about it. I don't want to put this in the chat because I really wouldn't know how to phrase it yet. But but something to think about for everyone who's on this: how often are you cleaning those other high touch surface areas that aren't in the operatories? And I'm talking about the front desk at the reception area. I'm talking about door handles and faucets, the refrigerator handles in your mini fridge and maybe in your reception area that has beverages in it, or even go back in your break room.
Hey, Dwight,
there, yeah.
If we think about it, we've got a slide. If you go up a couple more slides, we have a slide that shows the overall office. If you look at that, there's a cool thing on there. So I've always had resistance from my front staff, the receptionists. We don't need to do the bloodborne pathogen training. That's not our problem. We are not exposed. We don't breathe it in, and so on and so forth. Except if you think about, there's a patient who just had an extraction or maybe some perio, and they're holding some gauze in their mouth, and then they're going up to the front desk to write a check or to give them a credit card and things. Think about that counter up in the front, and think about whoever the reception is, whoever he or she is, has all kinds of exposure the same as everybody else. So the reality is, as we look at our offices, there's a whole lot of surfaces in there that we need to address and not simply bypass.
Yeah, I was thinking about that too. The real quick story. I went into an office because a lot of times I'll take these ATP meters with me, and you know, a lot of times people want to see it. And I went into an office, the very first, no patients were there yet. We were doing like kind of like a breakfast and learn, if you will. Cleaning crew came in there the night before. First thing I did was did the reception area countertop there, and their RLU count was into the 700s. And when I explained to them what that meant, they were kind of taken back by it. So it's really eye-opening. So again, you know, what gets measured gets done. We passed that slide up, but it was showing where a practice, you know, what it looked like before they started testing, and then what happened after they started testing and getting the results and seeing the trends. Obviously, when you see it and you know that you have an area of opportunity that you need to address, the trend was down on the RLU count, because now they knew about it, right? Knowledge is power. So they knew they had a problem where they didn't know they had one before, and they were able to address it that way. So, so just some considerations here, you know, reduce high touch areas. Talk about substances containing antimicrobial agents, additional protection for high touch surfaces, and the ability to treat non-porous and porous surfaces. So some methods that we can talk about real quickly. You know, surfaces are physically modified. Surfaces can be designed to inhibit the growth of pathogens. That could be altering material. Have you ever seen where, you know, products out there, consumer products, you know, are built in with antimicrobial? How do they do that? A lot of times they're using some metal ions into the plastics or whatever. Silver being one of them, copper. Those have always been proven as good antimicrobial agents. So they actually kind of built that into the products to be antimicrobial. Using chemicals, right, inhibits growth. And then there's some self-cleaning coatings, antimicrobial coatings and materials that we can talk about. And one of them is that's out there is called SprayGuard surface protection. And this is to clean high touch surfaces. You know, it's a process, right? So, you know, it takes approximately about 3 hours, and there's no patients and there's no staff. What they'll do, you know, they'll come in, they'll clean all the surfaces out, right? Remove the soil like we talked about. You got to clean it. You can't disinfect it if you don't clean it. So they're going to clean it. Then they're going to put on step 2 there, as you see, apply an EPA registered disinfectant. This one's called Smart Touch. Anyway, they'll apply that Smart Touch. That's your disinfectant, right? So you've cleaned it. Now they're going to hit it with this disinfectant, and then they're going to put a third application on there. And this chemical, which is part of the SprayGuard process, actually leaves a residual on there for 90 days. So it's going to continuously keep killing things for up to 90 days. So, and I think one of these slides here, this kind of really kind of gives you a good visual. If you could kind of picture a bed of nails, right? And these balloons falling on these bed of nails and popping, that's in essence what the SprayGuard is doing. And it's doing that up for 90 days. This is an adjunctive service. This is not to replace the current CDC protocols where we're cleaning and disinfecting. This is kind of the in-betweens, if you will. So, you know, if you think about it, if you come in and let's just use a reception area, for example. You come in and you've wiped that, you know, you've cleaned it, you've disinfected it. Doc, how long is that good for, you think?
You know, as we talk about all of these, everything is only as good as the first person who comes in. Next, the next person who comes in, unfortunately, we all have to breathe. We all have to exhale. Here we go. We now
Yeah.
have our first exposure. So to your point, when we look at this, this really works. And one of the things they've done as part of this application, they actually will use ATP meters as
Mhm.
soon as they've disinfected those surfaces. And then at the end of the process, they'll go back in and use that ATP meter again to show how effective this kind of treatment is. And the fact that it's got a 90-day residual is just about as good as you can look for.
Right, because it's continuing. Like you have your episodic protocols, right, where we're going in and cleaning and disinfecting. This is continuous. It just keeps going, right? You know, you think about overnight, or again, we talk about the other areas that aren't getting cleaned all the time, in the waiting rooms or in the break rooms even, and so forth. You know, there's a lot of restaurants that use this product as well. So it's there to help with the in between, but it is not there to replace what needs to happen. And in fact, you still need to clean the surfaces with this type of product. And when I was talking to the manufacturer, the reason why is when you have these microorganisms fall on top, and you know, again, you think of that bed of nails and those balloons popping, it's just like the cells, right? The cells fall on these spikes, it bursts that membrane, kills the cell. Then they'll accumulate on top, and then they'll cover those spikes. So that's why it's important to come again behind them and keep those areas clean, so those spikes can continue to work for you.
And Dwight, you use
Yep.
the term, it's great. You use the term adjunctive.
Yep.
This is an additional application. We still, in between every single patient, need to go in and wipe down all of those high contact surfaces with an N-listed disinfectant. Got to keep doing it. That never really goes away, and it doesn't matter what, when, or how. That still fits. Even in some of these hospital situations where you see these robotic devices with UVC and things, they still wipe down the counters to clean them, and then they still wipe them down with a disinfectant.
All right, let's talk about, so we've covered some surfaces. Now let's look at some airborne transmissions, and how we treat the air. So real quick, yes or, you know, yes, I have, or no, I haven't, in the chat. How many people on this webinar right now have installed a device, an air purification device, if you will, that is used to treat the air and kill airborne pathogens in your practices? I'd be curious to see, you know, who's implemented, and maybe even share what you implemented. Was it a HEPA filter? Was it just an air purification thing you bought at Walmart? So we'll let some people respond to that. So when we look at airborne transmissions, considerations include what size the particles are, how long can they remain in the air, and how far they can travel. So you've probably heard a lot, you know, we talk about spatter, we talk about aerosols, you know, what's the differences between them, how far can they go, how long. You know, when I first got into infection control several years ago, I was always taught that aerosols had a 4-foot wide radius, and were airborne for about 30 minutes. Now we've learned that's not necessarily the case. These things can travel far, they can stay in the air a lot longer, and a lot of it does have to depend on the size. So spatter is going to be greater than your 50 microns, right? Aerosols is going to be less than 50. And you know, you think of spatter, they're bigger, they're heavier, so they're not going to go as far, they're going to fall to the ground faster. Whereas aerosols, they're a lot lighter, so they can go a lot further. So here are some examples of just some airborne microorganisms here. You know, of course, flu, common cold, measles, TB, right? Legionnaires, and of course, the current pandemic we're in with the SARS, COVID. So reducing the risk. I'll take a look at this chat real quick and see. Got some Medify air purifiers, got HEPA with UVC, awesome.
So for everybody who responded that has a HEPA filter, it's imperative that you recognize those filters in general need to be replaced periodically. And periodically means, again, that's up to the IFU or MFU, manufacturer's instructions for use. Really important, because you're going to lose the efficacy of those devices themselves.
Yep. And kudos for you guys for doing that. You know, I kind of see it's kind of been 50-50. I don't know what you've seen, Doc, kind of around the country and the people you talk to, but I kind of feel like half people, well, maybe about 30% of the people have actually installed something for air, and maybe 70% haven't. You know, the CDC talks about taking a multi-layered approach to mitigating the spread of airborne pathogens. And of course, we can do that through some air purification systems. So those of you, you know, here's a little bit about HEPA filtration and how it works. They are recommended by the CDC and ASHRAE. They can run continuously, right? You're looking at removing 99.97% of particles. You know, the way the HEPA filters work, and, you know, I read somebody put in the chat that they had a HEPA filter with maybe like a UVC bulb in it. So, you know, those are what we would call our reactive, if you will, reactive devices. So in order for them to work, pathogens actually have to come through that filter in order for it to be captured. And then if you have a UVC, you know, for example, a UVC bulb that can eradicate that airborne pathogen while it's in there, assuming it hasn't pushed through fast enough. Because, you know, with HEPA filters and a lot of air purification systems, it's all about airflow and exchange rate, right? It's got to constantly keep turning that room, which a lot of times could be loud. And so I've talked to a lot of doctors who actually kind of turn it down, but then if you turn it down, it's not moving the air as much. But anyway, in order for it to work, those pathogens actually have to come through the device. So it's more of a reactive technology. And as Dr. Don mentioned, you know, make sure you look at those IFUs, you know, and make sure you replace those filters when they're supposed to be, so that it can work at its optimal level for you. So again, we talked a little bit just a second ago about, you know, ultraviolet. Ultraviolet's been around a long time as a way to disinfect. Here specifically UVC lights. And then you've got ultraviolet germicidal irradiation, the UVGI. So you can find these like in induct units. What's great about these is it does allow for kind of a continuous decontamination, right? I mean, whereas a HEPA filter or a lot of those air purifiers are reactive, it has to wait for it to come in through the device. Where if you have a device something like this, it's actually putting out the UV light and it's constant, right? It's safe, it's constant, you can be underneath it, it's constantly killing airborne pathogens. It takes a more of a continuous approach, if you will, proactive and a continuous approach.
Hey, Dwight, to your point, I think the key for all of us to recognize is that although episodic works, we wipe down our counters, we do all of that, continuous is the ultimate best way to do things. And we look at some of these approaches on this slide. Well, those robots that they have going into hospital ORs and things, realize they're all remote controlled, because you can't be in the room with that level of UVC. There is some modified UVC that can be installed in the ceilings in operatories that have been proven to be safe. So in those, they work. And likewise, if something is built into your ducting in your office, well, you're not going to be exposed even to very high levels of UVC, because it's going to be shielded, and then the air is treated within that. But again, wherever possible, I always look toward continuous rather than episodic. Second, somebody walks in or opens a door in your operatory, and most of our operatories are wide open, you've just eliminated your episodic approach. And if you don't have continuous, you're starting from scratch right from the moment somebody walks in.
Yeah, I mean, I used to talk about that again. I go back to dental unit water lines because I used to do so much with them. I mean, if you think about when every time you use that air water syringe, or every time you switch out the bottle, if you have a closed-ended chair with a closed water system, every time you've let oxygen into those tubes, you're letting in bacteria. And I, I remember OSAP boot camp a few years ago. We were looking at some of the testing that they had done, and the amount of bacteria and how fast it can grow in those dental unit, because those are very thin, right? Long, a lot of surface area in a warm, dark, moist place. I mean, that's where biofilm breeds and loves to be in. And so letting in just that little bit of oxygen, it can take off fast, and they can go right through those and grow right through those lines. So, that is, to your point, why it is important to have continuous, proactive and continuous, keep it going. Because I had a question, somebody had asked me about that, about the robots that you were talking about. Everyone's got to clear the room. They're gonna hit it with this UV light, and then once somebody walks in, now what? We just recontaminated the area again. So
Dwight, interesting. So when they talk about this, talk about HEPA filters, and then they talk to some extent about a HEPA filter with a built-in UVC light. So now you don't just collect, you collect and kill. The issue in my mind when I look at this is, what can we do to actually actively protect ourselves? And that's part of some of these other available devices out there. But the scary thing is, are they safe? Are they recommended? Sort of things that produce ozone. Do you really want to be breathing in ozone? Also, have some of those devices, as we see on this next slide. I'd go on to the next one because it tells an interesting story. We talk about, do we not want the FDA to determine the safety of that? And of course, we go back to that continuous versus intermediate. And what's the appropriate size for a device to protect us? Is it a small device? Is it a huge device? And we all want to know the data. We want to know what was really done, because it would be great to think we can protect ourselves and then put ourselves with a device that may even yield greater risk. So
yeah, and so there's a lot of due diligence that's involved with that, right? And that's what we would urge everybody to do is, make sure you do your due diligence with any device that you're going to be looking into, especially, but for any solution, that is going to be a main part. So so to your point, there are some devices that are out there. This one's called ActivePure. This one specifically is the ActivePure Medical Guardian. This is actually, as it says, they're utilizing space technology. It is FDA cleared as a Class 2 med device, so it is FDA approved. It is continuous air disinfection. They've got some different units. They've got plug and plays. They've got induct units they can use. It has been tested in healthcare settings, right? So
if you could go back to that for 1 second, if you would, Dwight. So if you'll take a look, the picture on the left is actually a picture of the space station. This actually exists on the space station as it is, which I love that idea, because that told me, well, in close confines, this is truly safe to be there. And yeah, there's all kinds of studies. You can do all the studies you want. That made me feel happier. And then I was looking at some of the other data on this particular device. They're using this in, I saw the Cleveland Clinic actually has this in each of their ORs, and now it's present in stadiums, in school districts. It's an interesting technology. And the device that ActivePure Medical Guardian is the one that has the Class 2 FDA as an actual COVID-19 kill. But in looking at the other devices, they have some small ones, a bookshelf size, actually, like bookshelf speaker size. and they all use the same technology that's in that Medical Guardian. I'm guilty. I have one of those in my kitchen and keep it on. The neatest thing I liked about that is they don't make noise, even when they're turned way up. And that was one of the key features, because we've always had HEPA filters in our house, but the reality is when you go to sleep, you got to turn them way down. They're noisy. So really does something.
Yeah, and that's that's one of the great things about this device. I mean, it's different in that it's actually putting out. So if you look at phase one there, it produces gaseous hydrogen peroxide, these hydroxyl ions and other oxidizers. And those oxidizers actually get into the air, get into the atmosphere, and it's destroying microorganisms. And you mentioned that, as far as this is FDA cleared as a class 2 med device, and it does have a SARS-2 kill claim. So it's actually going into the air and it's killing everything. It's on a seek and destroy mission. It's not waiting for those particles to come in and to be captured. That's irrelevant. It's going out and it's putting these molecules. So, air gets sucked into the device, then it takes the oxygen and the humidity, converts it into that gaseous hydrogen peroxide, and then it shoots it out and fills up the space. So it basically creates a protective bubble, if you will, right? You can walk into the space, you're in this bubble of protection, knowing that, as soon as things are coming out, airborne pathogens may be coming out, you're surrounded by these molecules, and it's just killing them.
Well, I think the coolest thing I saw in that is that within 3 minutes, you can have this total kill effectively safe in that square footage that they're rated at. So as I said, I think that going on to this next level of technology, and for all those offices that did invest in the HEPA filters and things, this is truly, I think, the next level of technology that will be utilized. Because remember, this is producing these out into the air. You don't need to suck the organisms through a loud fan or whatever. It actually is out there surrounding you.
And even though we talk about SARS-2 and COVID right now, right, there's still a lot of other airborne pathogens that exist. I mean, as we saw on a few slides back, the flu's still out there. It's still around. You don't hear it as much, but it's there, right? And there's other things that you can catch that are airborne. But then you got to think about, like right now we're talking about COVID-19. What's to come a year from now, 2 years from now? Now we've got all these different variants, right? So it's going to continue to keep going. And that's why something like this kind of a device can just kill things. It can go out and kill, it's killing the microorganisms.
And it was really cool because they actually tested it on proxy, similar bacteria to like MRSA and the organism of Black Plague. And they tested it on similar organisms. They call them proxies. And on those, the kills were phenomenal. And that's what those charts show. So this is a product that's had independent testing. I know they did a bunch of testing at University of Texas, which should please you, obviously, with your accent, Dwight. I didn't hear anything on them doing anything at NYU for me, but that's okay. So the reality is these have really been well tested, super safe, and they work.
So some of these slides here, we're just, you can see some of the studies for yourself. Take a look at them, what they did. And you can see the time that it took. So another device that's out there is called the Radic 8. They were first developed in South Korea during the SARS outbreak. And then it's got 2 main phases, right? Air gets sucked in. It's got the HEPA filter and an activated carbon filter. Puts out some, uses some oxidizers to kill while it's in the device, and then the clean air leaves the device. So that's more of what we would call a capture and kill that we were talking about earlier, Dr. Don, where it's going to come in, it's going to try and eradicate it inside the device and then push the clean air out.
Yep.
So, in summary here, contaminated surfaces pose a risk. We all know that. CDC recommendations we talk about for cleaning and disinfection of clinical surfaces, they obviously should be followed. Again, not to beat a dead horse, but I'm going to beat a dead horse. Follow the instructions for use, follow the instructions for use, follow the instructions for use. Those are very important. Please take the time to read those. Seriously, I've been in a lot of dental practices, too many to count. And we look at the IFUs, and a lot of times, and I'll tell you what happens, Dr. Don, is, well, I didn't know that, right? I get the answer, well, I didn't know that. Well, that's not how I was taught. Well, the problem is if the person who was teaching you didn't learn it the right way, then they're teaching it the wrong way, right? And it's just that cycle that goes on. Take the time, look at those instructions for use. If you have questions, ask your distributor, ask the manufacturer. It's really important that these solutions, whatever they may, they do what we intend them to do. You know, otherwise, what's the point?
So Dwight, before we end, I'm going to ask Marcy, who is in the background, who's been keeping track for us. Has anybody asked what that is over my shoulder? Because I find that hurtful, that you could be looking at what could be an incredibly good dessert and nobody's asking what the heck it is.
Nobody has asked just yet, but they may now.
Yeah.
Okay, so I will cheat and give the answer. The answer is, it's called an affogato, and it's an Italian dessert with vanilla ice cream in espresso.
Ooh, I've never had one of those.
Somebody said it looks like a turkey. You're
talking about you.
Me, yeah, that's what I figured. You're not talking about dessert. That's about me. I get it.
Nice one, Holly. Thank you for that. We talked about ATP meters again. This is a really great idea. As I said before, what gets measured gets done. If you don't really know, find out, right? That's an objective test. It's not subjective. We talked about some adjunctive surface protection solutions that you should look at incorporating into your practice. Again, it's the continuous versus the episodic, the in-between stuff. We talked about air decontamination. Airborne microorganisms can be transmitted, causing disease. We talked about aerosols, reducing those, some CDC guidelines. Recommended adjunctive devices included HEPA filters, UVGI. And guys, do your due diligence, ask. We're always available for questions. We would love to help in any way. There's a lot of knowledge with Dr. Don here. We're super excited that he's on the board of directors of OSAP. He is on the medical advisory board. He is our the chief clinical officer, and he's got a lot of knowledge there because he's got a lot of years.
Knew that was coming.
Once again, everyone is going to receive an email by the end of the week with your CE and a link to get all that. So just know that that's coming. If there was anything that we didn't get to in the Q&A or in the chat, we can respond to those as well. Thanks, guys, for joining us today. We hope you got something out of this. And Dr. Don, thank you for being here today. I really appreciate that.
Thank you, everybody.
All right. Happy holidays, everybody. Thank you.